Okay, before we started talking about photosynthesis, I wanted to give you some background information. So we're going to start big, and then we're going to get closer and closer and closer, and smaller structures. So first of all, looking at something really big, history of life on Earth. So up here, these are billions of years ago. So we can see the first prokaryotes arrived about 4 billion years ago, or we have fossil evidence of them. And the eukaryotes came around 2 billion years ago. And what happened in between those 2 points? First of all, our atmosphere did not used to have oxygen. So all of the prokaryotes that were living on the earth, they were all anaerobic, facultative anaerobes. Some would actually die in the presence of oxygen. So at some point between these two, we had photosynthesizing bacteria that started creating oxygen. And so once the oxygen started accumulating in the air, it would have become toxic to those bacteria that had not evolved the ability to use oxygen. So they would have started to die off and everything would have been shifting. We talked about the endosymbiotic theory. Perhaps those facultative anaerobes engulfed some photosynthesizing bacteria or some aerobic bacteria and were able to utilize the oxygen, or they were able to make their own food. So oxygen begins to accumulate sometime between 2 and 3 billion years ago. And today, all organisms on Earth require energy, and most require it through cellular respiration using oxygen. There are still some facultative anaerobes. So we talk about autotrophs. Those are those that can make their own food from things in their surroundings. And usually we're talking about photoautotrophs, which are using sunlight to make chemical energy. And then some are chemoautotrophs, where they're using other inorganic compounds to make organic molecules. So I think that we would typically think of autotrophs as being plants, but we're kind of limiting our thoughts when we do that. We want to remember that they come in various kingdoms. So we do have the cyanobacteria in the bacterial kingdom. We have a number of photosynthetic protists. Algae is a form of protist. Euglena have chloroplasts in them as well. So when we talk photosynthesis, more than just plants are accumulating oxygen in our atmosphere. But we're going to talk about plants. So again, we're starting big. What are the major needs that a plant has? It needs to collect light. It needs to store the light. It needs to use that light to make building blocks. And those building blocks it needs to make bigger molecules. So we're going to be talking about leaf structure here in a little bit. So we'll talk about where they're collecting light and storing that light. And then down here, these are some elements that plants absolutely need in order to assimilate the molecules that they need to build pieces and parts. So we know that carbohydrates, lipids, proteins, nucleic acids all require carbon, hydrogen, and oxygen. We know that nitrogen is needed for nucleic acids and proteins. We know phosphorus is needed for nucleic acids and phospholipids that are in all cell membranes. We know potassium is needed for the sodium-potassium pump. We know sulfur is needed to make proteins. And magnesium we haven't talked about yet, but it is a component of chlorophyll. So we're really going to need magnesium if we're going to be able to photosynthesize. So some common questions that you see on the AP exam have to do with assimilating molecules or tracing atoms. So first of all, you might see questions, and whether it's photosynthesis, cellular respiration, any chemical reaction they're talking about, they could throw a tracer in there. And so they might put a radioactive isotope of oxygen or a heavy carbon, and they want to follow that element. So when you're analyzing those questions, you want to think, where would that element typically go? That's where the tracer is going. So the heavy carbon is going to just take the place of a normal carbon, and wherever it goes, a normal carbon would have went as well. So maybe we trace carbon dioxide, and we see that carbon ends up in glucose. We trace the oxygen, and we find that the oxygen from carbon dioxide does not end up as oxygen. We traced water and we find the oxygen from water turned into oxygen. So that's what we use tracers for, and you'll see many questions similar to that. Other types of questions related to chemical reactions is when they take a component out and they ask what could not be made. So if you were growing plants in mediums that lacked one of these items, you were growing plants that lacked phosphorus, for example, what could it not make? Well, it's in phospholipids and it's in nucleic acids, so if you don't have it, you can't make either of those. So you'll see questions like that where they'll take one of these elements away and then ask you what that absence would lead to. So plants, how are they going to get the things they need from their environment? Bacteria are cool, they can just absorb it through their cell membranes, but plants are going to get it for the most part through their roots. So they're going to uptake water through their roots. We've talked about water potential. They're going to take in nitrogen. Some plants have nitrogen-fixing bacteria living right in their roots. So we talked about soybeans before, and they have those rhizobia that live in their roots, and they have that symbiotic relationship. They're going to take phosphorus, potassium, sulfur, magnesium, iron, all those things, and they're going to take it into their roots so they can assimilate them into larger molecules. The stem, we know that the stem has the xylem and the phloem in it, so the xylem is going to move water and the phloem is going to move food, and we know that water will continually move from its highest water potential to its lowest water potential, which is at the leaves. And then we have our leaves, and that's where we're going to do gas exchange. So kind of like us breathing in oxygen for cellular respiration and breathing out CO2, plants will also respire. They will take in oxygen, they will get rid of CO2, but they also photosynthesize. So they're going to take in CO2 and they're going to get rid of oxygen. And some of the oxygen stays in there and they breathe it in and breathe it out, so to speak. They'll use it for cellular respiration and they'll produce it from their own photosynthesis. So gas exchange is taking place in little holes called stomata. So like I said, I want to continue getting smaller and smaller and closer to detail. Okay, looking at a leaf structure, the leaves have the upper epidermis, kind of like our skin. We've talked about this before, that it has a cuticle and usually a waxy substance that protects it. You have the palisade, you might say the palisade layer, the palisade chlorophyll, the palisade mesophyll. Different resources will use different terms, so the palisade layer is what I'll say. Below that, the spongy mesophyll. We can see a little bit more spaciousness there. You have the lower epidermis, and then under, on that lower side of the leaf, typically is where we find our stoma or stomata. So over here, we're zooming in on the stomata. The stomata have two guard cells, and the pressure of the guard cells fills them up or releases them. And so the guard cells can open and let gas exchange occur, or they can close and make it so gas exchange can't occur. So air is going to come in through there. We're going to use CO2. We're going to use a little bit of the oxygen. So that is how the plant might be breathing, if you will, but it's bringing in the CO2 that it needs for photosynthesis, and that's what we're concerned with right now. Most of the photosynthesis then is actually taking place in the palisade layer up here. So there's lots and lots and lots and lots and lots of chlorophyll in the palisade layer, so the CO2 will go up into the palisade layer and be used for photosynthesis. So deeper and deeper we're going. Let's go into that palisade layer, and in the palisade layer we find our chloroplast. So chloroplasts, we can break those down into even smaller details. So the chloroplast has a double membrane here, and then on the inside it has stacks of thylakoids. So an individual disc is called a thylakoid, the whole stack is a grana. This is where the chlorophyll is. So this is where light-dependent reactions occur. This is where we're absorbing light. Outside of this, in the stroma, the space here is called the stroma. So to me, I try to make it analogous to cytoplasm, the cytoplasm of a cell, the stroma of a chloroplast. And out here is where the Calvin cycle will occur, or carbon fixation. This is where we're going to make our organic molecules. So now I'm going to take one of those thylakoids and I'm going to blow it up. So here is one of those thylakoid membranes inside, and what do I see there is a bunch of hydrogen ions. So what makes the formation of ATP possible is a high concentration of hydrogen ions. So I need to keep a gradient between the two sides of the thylakoid. So remember, hydrogen and any other thing is going to move from its high concentration to its low concentration. So we don't want this to ever reach equilibrium. If it reaches equilibrium, diffusion will really slow down and we won't make nearly as much ATP. So we are going to use a hydrogen ion pump. Pump, 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 pump. We're going to push hydrogen in there. Many of the hydrogen are coming from water molecules. So the hydrogen we're forcing into a highly concentrated area already. So it's a great example of active transport that keeps the concentration high so that there's always a gradient, and then hydrogen can keep flowing through the hydrogen, the ATP synthase. So that will make ATP that will be used in the Calvin cycle out here in the stroma to make organic molecules. So we're going to continue on through the thylakoid. In the thylakoid, we have clusters of pigment molecules. Those pigment molecules are chlorophyll. So we're typically talking about chlorophyll A and chlorophyll B when we're talking about photosynthesis. And these are found within the membrane. Right there you see that magnesium I referred to earlier. So plants need magnesium as it's a component of photosynthesis. And a number of these molecules all in a group is called a photosystem. And there are two photosystems that we talk about. Photosystem 1 over here is going to absorb the wavelength 700 nanometers. Photosystem 2 over here absorbs the wavelength 680 nanometers. So this is a cluster of pigments, and so you call it a photosystem. So we typically call this photosystem 1 and photosystem 2. Now, photosystem 1, photosystem 2, sometimes kids are confused because they see this arrow going from 2 to 1, and so they want this to be the other number. The reason why this is photosystem 1 is because it was discovered first. That's all. So that has nothing to do with the order or the flow of electrons. Just its sequence of discovery. So we can see quite a bit of the wavelengths that are absorbed are going to be in the red and in the blue spectrum. So this is called a light absorption spectrum. You might see a graph like this that you have to interpret on the exam. So chlorophyll A, chlorophyll B, and carotenoid. Carotenoid is one of a number of accessory pigments. So plants have more than just chlorophyll that allow them to absorb wavelengths of light. Remember, pigments produce color by reflecting certain wavelengths. Here we can see they're not absorbing any green, which is why plants appear green, because they're reflecting green wavelengths. So pigments will absorb certain colors of wavelengths of light, and then they reflect the other wavelengths of light. And these are all kind of in the same range, but by having a number of different pigments, they will be able to absorb more wavelengths of light, and so it's more efficient for the plant. So that's, I think, all of the background you need in order to be able to start talking about photosynthesis and not get caught up in the structures. So thanks for watching.